Our final speaker, last but not least, we have Ryan Krantz from Loyola University, Chicago.
All right, you can see that okay?
Looks great, yeah.
All right. I'm a master's student in Dr. John Kelly's lab, and I'm happy to share today some of our recent findings regarding the interactions between triclosan and microplastic-associated microbial biofilm communities. So triclosan is a broad-spectrum antimicrobial compound that is a ubiquitous additive to plastic resins for use in things like textiles and other consumer goods like kitchenware and things like that. As well as personal care products. So it is a common contaminant in wastewater especially, and can mix with microplastic in that context. We are interested in when triclosan adsorbs to the microplastic surface, whether it has any effect on microbial assemblages and diversity. We use a microcosm experimental approach to test this, wherein we initially took fibers from 3 polymer types, seen at the bottom there, acrylic, nylon, and polyester, cut from thread, and then exposed them to stock triclosan solution for 24 hours to give it ample time to adsorb to the microplastic surface. We measured the rates of adsorption, and this is actually a metric that we haven't investigated as much yet. But interestingly, the different polymers did not adsorb triclosan to the same extent. After this, we created replicate samples of fibers exposed to triclosan and also microcosms with fibers that were not exposed to act as controls, and then incubated them for 30 days at 80 RPM, along with water sourced from a local river to introduce environmentally relevant microbes. So our initial findings seen in these beta ordination plots indicate that there was a significant effect of exposure to triclosan on the microbes, both on microplastic samples and the water. So to kind of orient you here, I have bacteria and algae, but the color codes and the keys are the same. So we have initial pre-incubation samples, and you can see a shift both in the water and microplastic in the top left of both of these plots, as well as a shift in the opposite direction in the control samples, both in the water and microplastic. These ellipses are 95% confidence intervals. So any gaps that we see are significant differences in diversity and assemblage. Overall, you can see a pretty clear gap, and especially the control samples and the triclosan samples in the algae. We took these findings and then further investigated them by composing abundance plots here. So again, to orient you, there's absolute abundance on the top row, and then on the bottom row, proportional abundance, where every rectangle of a different color is representative of one of the top 20 most abundant bacterial or algal orders. So on the bottom here, if you look at changes from control to triclosan per polymer type, we see that formerly, or rather orders that are particularly abundant in control samples were dramatically decrease in abundance in triclosan treated samples. We see this in bacteria, but we also see a coinciding increase in some previously not very abundant orders, which is interesting. But the same trend extended almost to a more dramatic effect with algae. So here we see in microplastic and water, respectively, 2 orders that pretty significantly shrank in size, and then a very dramatic increase in the abundance of other samples across both microplastic and water. These findings are significant in the context of pollution of microplastics, considering the ability of microplastics to absorb other pollutants like triclosan, and then carry them through natural systems where they could interact with natural populations of microbes and potentially affect microbial ecology in those in those areas. But thank you for your attention, and I hope everyone has a nice evening.